Okay, at the end of podcast part 1, we introduced the polysaccharides, starch and cellulose, and the fact that humans can digest starch but can't digest cellulose. Well, how is this possible? Well, I want you to get in your notes that starch stores glucose molecules for energy, while cellulose is used in plants as a rigid molecule for support. So while one is an excellent source of energy, the other is used for making paper. The difference is in their structure. Write in your notes that plant starch is made up of amylose, which uses the enzyme amylase to break down into maltose, which then uses maltase to break down into the monosaccharides glucose and maltose, all of which are naturally produced by mammals. Not naturally produced, but naturally digested by mammals. Plants, on the other hand, use the enzyme cellulase to break cellulose into a substance called cellobiose, which then requires the enzyme cellobiase to break it down further into glucose. Humans do not produce the enzyme cellulase nor cellobiase. Those enzymes are produced by the second and third chambers of the stomach in other mammals such as cows. Make sure you have that written in your notes. Okay, enough about carbohydrates. Let's move on to an extremely important molecule, proteins. Remember, proteins are often referred to as polypeptides and are organic compounds made up of amino acids bonded together with peptide bonds. Organisms simply cannot function or survive without proteins because they are essential parts of organisms. Write that proteins make up half the dry weight of a typical cell, and that the reason proteins have such diverse functions is due to their ability to bind other molecules specifically and tightly. Let's get some functions and locations of proteins in your notes so you have many examples to use on the exam. So they can be structural, which means they make up bones, skin, nails, hooves, and hair. So you should probably get some of these in your notes. They're enzymatic. They act as enzymes is what that means. Enzymes digest sugar, make DNA, make fatty acids, and many other functions that we won't mention all here. They function in transport. For example, they carry oxygen and fats in the blood. And we'll talk about calcium and chlorine, the calcium-chlorine channeling later in this class. They can work as contractiles, in other words, muscle fibers for movement. We'll see that here in a second. Or they can move chromosomes. They can function as a hormone, for example, to regulate blood sugar or increase heart rate. They can function in immunity. Their antibodies fight foreign substances are made of proteins, for example. They can be pigments. Pigments in skin, eyes, leaves, etc. are proteins. And they function in recognition on cell surfaces, for example. When we talk about cells, we'll talk about how those proteins function. They can also function as receptors. And they can function as toxins. In other words, they can stop nerve transmission, affect movement of ions, and enzymes that destroy red blood cells. So, man, that is a lot of functions. Proteins, extremely important. Now let's look at the basic structure of a protein and get some of these structures in your notes. Now, I'd like you to get this labeled drawing in your notes first, so do that right now. As you can see, proteins, also called polypeptides, are composed of 2 or more amino acids bonded together with a peptide bond. Now, in this diagram, you see a single amino acid. So you see the amino part of it over here that you'll recognize with the nitrogen center and 2 hydrogens hooked on. And you'll see the acid center here on the right-hand side where you see a hydroxide here and a double bonded oxygen. You also see a central carbon atom. And then something called an R group down here. So make sure you get those all in your notes, and they're labeled exactly like they are in this diagram. So let's talk more specifically about the R group once you have this diagram in your notes. Now get in your notes that there are 20 different amino acids found naturally out there in proteins, and that most microorganisms and plants can biosynthesize all 20. Now remember, the word synthesis means to put together. Also write that it's the R group that distinguishes these amino acids from one another. So it's this R group that distinguishes each amino acid from the other. Now it's worth repeating that most microorganisms can produce their own proteins. Contrast that with animals, including humans, who must obtain some of these amino acids from their diet, in other words, the food we eat. So write in your notes that the amino acids that an organism cannot synthesize on its own are referred to as essential amino acids, and that humans require 8 essential amino acids. These are amino acids they can't synthesize themselves, where other organisms can. Those are called essential amino acids. By now you should know dehydration synthesis, but it's worth getting into your notes that it's also called a condensation reaction. Can you figure out why? Now the reason we want to mention both is because I've seen both used on the exam. So remember that a dehydration synthesis is also called a condensation reaction. You see it right down here. Now, I ask you if you thought you could figure out why. It's because a molecule of water is removed from 2 glycine amino acids to form a peptide bond. We've seen this diagram earlier in the podcast. So one donates a hydroxide while the other donates a hydrogen. And those two together form a water molecule. Remember that a protein is called a polypeptide. So on the right here, you see a protein made of 8 amino acids. Now the prefix for 8 is octo. So this is an octapeptide. In other words, there's 8 amino acids in this protein. It's called an octapeptide. It's also a polypeptide because it's more than one. Okay, now we're working off the idea that there are 20 naturally occurring amino acids. So it makes sense that there would be many, many different combinations possible that would make up a protein. So you could combine those 20 amino acids a lot of different ways to make a lot of different proteins. Get in your notes that the order in which amino acids are bonded is called the sequence, and that the sequence of amino acids determines the primary structure of a protein. So make sure you get that down. Also write that the primary structure of a protein is determined by the genetic code, which oddly enough is the sequence of polynucleotides in an organism's DNA. Make sure you get that down as well. So the primary structure is the sequence of amino acids in a protein, and that's determined by the sequence of polynucleotides in an organism's DNA. We're going to talk about protein synthesis way more specifically than this later in the class, but for now, understand it's the sequencing of amino acids that determines the protein. That's called its primary structure, and that's determined by the sequence of polynucleotide in an organism's DNA. Okay, back to that R group again. Write in your notes that it's the chemical nature of R groups that defines the characteristics of a protein. So it's the chemical nature of the R group that determines the characteristic of a protein. So what does that mean? It means 2 things that we want you to get in your notes. 1, Some amino acid groups are attracted to each other. However, some amino acid groups are also repelled by each other. Write down that amino acid groups determine the overall shape of a protein because proteins are not long, straight molecules, but are usually folded into a 3D shape like you see here in this slide. Can't be 3D because it's not a 3D picture, but you kind of get an idea. Here's that primary structure with all these little amino acids that are peptide bonded together. And then what starts to happen now is this starts to change shape. And in the end, you get this convolution now called quaternary structure that we'll talk about here in a second. But if you could see it 3D, it would be like a big ball, a 3D ball of all these convolutions in here. There are actually 4 levels to protein structure we need you to write down. The primary structure we've already mentioned, which is the amino acid sequencing. Now, secondary structure, that's right here. Here's your secondary structure. So here's primary, here's secondary. Secondary structure is when this sequence of amino acids are linked together by hydrogen bonds to form 2 structures, pleated sheets, right here, and alpha helix. I don't know what the plural of helix is. It's helixes, I guess. Pleated sheets and alpha helixes. So we get this primary structure that now breaks down into a pleated sheet and an alpha helix. In the tertiary structure, here's your tertiary structure, folding begins like you see here due to attraction between these sheets and helices, there we go. And then in quaternary structure, down here, here's the quaternary structure, several amino acid chains are folded together like you see here in the bottom of this slide. This sequencing of folding all leads to the different functions of a protein. So I'd like you to get this entire diagram in your notes. Now, I know you're not artists, but this will help you to get an idea of what things look like. So primary structure is the sequencing of amino acids, breaking into pleated sheets and alpha helices, I guess that's the plural. Then these start to fold as they're combined. And then this quaternary protein structure consists of more than one of these amino acid chains. And they're also folded together. Very complicated structure. Okay, now let's briefly mention structural proteins and how they differ from a normal protein. So we're going to talk about structural proteins and kind of set them apart from normal proteins. Write in your notes that structural proteins confer stiffness and rigidity to otherwise fluid biological components, and that most structural components are fibrous proteins. F-I-B-R-O-U-S. Fibrous proteins. So most structural proteins are fibrous proteins. Now, also write in your notes that these structural proteins polymerize. There's a word, polymerize. You should understand what that means basically from our talk of monomers and polymers. They polymerize to form long, stiff fibers that comprise the cytoskeleton. C-Y-T-O, skeleton, the outside skeleton, in other words, which allows the cell to maintain its shape and size. Now, a good example of a structural protein are actin and myosin, and you see them here on the left. Actin and myosin are muscle proteins. You can see an example of the structure over here on the left, and these now work to have muscles contract. Actin and myosin. Another example would be the protein collagen, right here on the right-hand side, which is found in your skin. You see the secondary structure, the tertiary and quaternary structures of collagen here. So collagen would have a completely different structure, so it would have a completely different function, these actin and myosin fibers. So the folding would be different. So although these molecules look similar, quaternary structure, they wouldn't be exactly the same. Also, it pays to write this down again, that structure is directly related to function. So if the folding is different, so I'd like you to write this down, so if the folding is different for each protein, it makes sense that the functions of each protein would change as well, which is shown here in this slide. You see many, many different structures here. These would all be proteins now that function differently. So where does the information for folding of your proteins come from? If you said your DNA, you would be correct. And even more specifically, the DNA that you get from your mom and your dad. Okay, that's it for part 2, our discussion of proteins. So go ahead and submit those notes to Moodle after you take a picture of them. Pick up that study guide, do those questions, and submit those as well. And then we'll see you soon in part 3 of our discussion of biomolecules. Bye.